This is an image of mostly hyaline cartilage. There is some bone here and collagen here. The extracellular material, or more specifically carbohydrates in the matrix of cartilage, contains lots of ionized sulfate groups, so exhibits basophilia. It's reacting with the basic dye, and that's hematoxylin. This is grey matter in the spinal cord. All these nuclei are stained with hematoxylin because the heterochromatin and also the nucleoli contain ionized phosphate groups in the nucleic acids. The granular endoplasmic reticulum in these large motor neurons and also ribosomes contain ionized phosphate groups on the ribosomal RNAs. They also exhibit basophilia, so they're going to stain a bluish-purple colour with the basic dye of hematoxylin. And so the cytoplasm often has this bluish tinge, or more apparent in these neurons as bluish masses. It's hard to see the endoplasmic reticulum and ribosomes in many other secretory cells when they're making protein components, which would be eosinophilic and therefore totally dominate the cell cytoplasm. Acidophilia, or as I said earlier, when we use eosin, it's sometimes referred to as eosinophilia, can be demonstrated in a number of different tissues. For instance, in the upper half of this image are smooth muscle cells. Acidophilia, or eosinophilia, is exhibited by the contractile filaments or contractile proteins inside these muscle cells. So you would expect cytoplasmic filaments in skeletal muscle fibres and cardiac muscle fibres or cardiomyocytes to also exhibit acidophilia. In the lower half of this image are extracellular fibres of collagen. The ionized amino acid groups exhibit acidophilia and stain this pink stain with eosin. Red blood cells are also acidophilic. Now here's an example where you can see both stains used together to distinguish typical cell components. Again, like I've mentioned before, you will come across in most of your histological sections. This image is a section through the stomach. You can see a number of different staining features here. For instance, on the very surface, you see these invaginations of an epithelium. And those invaginations have clear, staining mucus cells on the surface. The mucus stored in the cell cytoplasm was lost during the alcohol dehydration of the processing, and therefore there's nothing there to stain, so it appears very clear. Down further into the tissue, you see a population of pink stained cells and a population of bluish stained cells, with a few pale stained cells in the middle. They're cells that are exhibiting acidophilia and basophilia respectively, staining with eosin or hematoxylin. The very dark pink stained cells, called parietal cells, exhibit acidophilia or eosinophilia. They're eosinophilic. They're reacting with the eosin dye. And a lot of reasons why these cells are staining a bright pink is because they are full of mitochondria, which are eosinophilic or acidophilic components of cells. And those mitochondria are very important in producing hydrochloric acid, which is a product of the stomach. The more bluish stained cells down the bottom are exhibiting basophilia. Their nuclei stain a very dark purple colour, and here you see that the cytoplasm, unlike the red stained parietal cells, is stained pale blue. They're chief cells of the stomach. They're synthesizing pepsinogen, and that requires an enormous factory of granular endoplasmic reticulum and ribosomes. And if you remember back a few moments ago, I said endoplasmic reticulum is highly basophilic. And you see it clearly here because it's dominating the cytoplasm of the cell. Now, just one final point about tissue processing is that within this image, you can see little strands of whitish areas. Normally, that's interstitial or intercellular space. But it's a little exaggerated in this section, and it's exaggerated in a lot of hematoxylin and eosin stained sections. And that's because when you cut a thin slice of tissue and pass it through alcohols for dehydration, it often gives rise to differential shrinkage of different tissue components, resulting in what we see here as false spaces. They don't really occur in the real life of the specimen. They're artifacts. So now I think you should be familiar with hematoxylin and eosin staining. Next, I'm going to describe some of the limitations of using hematoxylin and eosin stains.